How many plants should be?
Oh, I thought you might like to point.
Observed in the-
How many plants? Well, that's a good question for a number of reasons. So when we are looking at differentials, right, our differentials are homozygous. They're like a variety. So if our differential is pure, we should have 1, 5, 10, 50 plants that all look like that. It doesn't matter how many you test. It's a fixed genotype, right, like a variety. There's no genetic variation within a variety. There's no genetic variation within our differentials. So all the plants should look like that. All right? So I'll just make a further comment about that, okay? So all the plants should look like that. Now what happens sometimes is they don't. And that's the reason for that is that somebody has made a mistake when they've been growing the lines in the field, and when they've harvested them, they've mixed them accidentally. And it happens all the time. It happens in my lab. And the problem is when your differentials get mixed, when your stocks get mixed, if you don't know the genes, you don't know which component is right and which component's wrong. So it's very difficult then to unmix them, right? So that's not genetic segregation. That's just due to mixing, admixture. That's a mistake made in increasing lines in the field. People do it all the time. So sometimes you will see different infection types within a differential. That's not due to genetic segregation. That's due to admixture or contamination of the seed.
Robert, you may also mention that infection can fail.
Infection can fail, absolutely. That's a good point, Zach. So you need a susceptible check in your differential set. So you could take a rust isolate from the field and you want to know what pathotype it is, and you inoculate your differentials and every single one of them is resistant. And you say, oh, this is B, 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 B by the North American system because it's low on everything. Well, it may be that it failed. It might not be avirulent at all. The rust is dead or the infection hasn't worked. So you need to have in your differentials a susceptible control, and if there's no rust on the susceptible control, you've got a problem with your inoculation. You've got to do it again. All right? Counting the number of plants is when we get into the genetics, when we're making crosses and we're looking at the progeny and you see different responses. And then you need to count the number of different plants. But that's when you have a cross, a population derived from two parents. This work is being done with fixed cultivars that are genetically uniform.
But usually in a differential set you would use between 4 and 10
4 and 10 plants. Yeah.
Per differential.
Correct. Yeah. So we would plant, you know, between 4 and 10 seeds, maybe a dozen seeds of each, just to make sure we get at least a few nice plants that we can read the infection type on.
When you have X type of disease reaction, then it is high or low?
It's low. Yeah. People know, I didn't really go into detail on that, but do people know what the X infection type is? There is a picture here somewhere. I can show here. All right, so this is the Nort. This is the immune one. The Fleck just has these little chlorotic spots. The 1 infection type, you tend to have a small pustule with some dead tissue around it. The 2 is the classic green island. The 3, you have largish pustules but some yellowing around them. But then interestingly, sometimes you get this X infection type where you get all of those. You get the Flecks, the 1s, the 2s and the 4s all on one leaf. And we consider that to be an incompatible interaction, a resistant reaction. And it's very typical of some resistance genes. So in stem rustic, before I gave the example of SR17, when we're doing a test with a culture with a pathotype that is avirulent on SR17, we see this X infection type. So then we know the pathogen is avirulent for SR17. And I'm really going to be really interested one day when somebody clones one of these X infection types and tries and explains to me why we get that. It's a puzzle. We don't know why. It's a really, really strange phenotype. But it is definitely incompatibility.
Same scale for stem rust and leaf rust?
We use the same 0 to 4 scales, but the infection types between the rusts are actually different. The actual phenotype, I should say, is different. All right, so on average with leaf rust, we would see similar kinds of infection types to leaf rust as we do with stem rust. But with stripe rust, the different categories are they're still 0 to 4, but the phenotypes are very, very different.
Maybe mention the fact that when you test barley seedlings, most of them will be X.
With stem rust. Yeah. Yeah, barley's a very, very odd thing. You can test hundreds and hundreds of barleys, and they are, for all intents and purposes, susceptible in the field, but they all look like this, right? Now, I know that sounds like I just contradicted myself because I said this is a resistant response, and yet we see in barley this resistant response in seedlings, but susceptibility in the field, all right? But that's a really important point. What we're doing with these seedling genotypes that we're working with, we're not doing this work, we're not breeding wheat varieties for people to grow as seedlings. Farmers need to grow these things to maturity. So throughout the work we do, we always have to correlate that seedling response with the field. We're using the seedling phenotype to make a prediction of what's going to go on in the field, and sometimes that doesn't hold up, and in barley it certainly doesn't. So that we've always, we can say, okay, we can do all these tests in the greenhouse, we can select and everything else, but the ultimate test is in the paddock because it doesn't always correlate.
How easy is it to separate an X from a mix of races?
Honestly, if you only have one line, very difficult. All right, so Dave's question is, this looks like we could have 3 or 4 different races in here, some of which are virulent and some of which are avirulent, and that's true. It looks just like a mixture of pathotypes with differing pathogenicity on a resistance gene. If you're only looking at one line that gives you the X infection type, I'd say it's almost impossible to separate an X infection type from a mixture of pathotypes. But if you have other differentials in the set, often you'll pick up the mixing on those other, the several pathotypes on those other differentials. But in the absence of those other differentials, almost impossible. What I normally do, the approach that we use, I'm not sure, I think, Zach, you start with single pustules, don't you, in your race analysis?
No.
You don't?
Start with a bulk.
Okay, so in North America, when a sample is sent in for race analysis, they will always establish single pustule isolates before they go to a differential set. So they end up with an isolate that's pure. We don't do that. We get a sample sent in from a farmer's field, we just mix it up and put it on a differential set. So we might have 1 pathotype, we might have 2, we might have 3. I've even seen 4. So how do we work out what they are? Is it an X infection type? Is it a mix of pathotypes? It's difficult. So what we do is and we'll talk a bit about this, I think, in Kumultar on the weekend, but what we do is we take out these single pustules. So we would take that pustule out and increase it on a susceptible. We take this one out and increase it on a susceptible. We might take that one out and increase it on a susceptible. So then we would have 3 single pustule isolates and put them back onto a differential set, each one onto a different differential set, to see whether or not they are in fact different pathotypes. So we do subculturing of single pustules to try and figure out those mixtures. I was talking to Zach about this yesterday. Just one final comment. You look at this and you think, oh yes, different infection types. I absolutely love doing this work, and Zach, we were talking about this yesterday. You could put me in a greenhouse with plants and rust on them and ask me to phenotype them and I'd be happy for weeks. And I think, Zach, you were saying the same thing. These infection types are absolutely beautiful to look at, and when you learn to read them and you know the genes, just by reading those infection types and using the gene-for-gene hypothesis, you can tell a lot about the underlying genetics.